Sorry guys, I ran out of time on the last video, so we're going to continue on with the second video. So the type of diffusion we just talked about with osmosis and simple diffusion is in this diagram here. That is when we have an area of high concentration. Those things are small enough to be able to fit through the membrane, so they're going to pass through here, and they're going to move to an area of low concentration. And that is going to continue to happen until we reach equilibrium, where the number of molecules on the outside equals the number of molecules on the inside. Now, facilitated diffusion, that is when we have a high concentration and we're moving to a low concentration. We do not need energy, but these molecules are just slightly too big to pass through, so we're going to use one of our integral proteins to do that. Those molecules can fit through this channel and pass through. Okay, so those are the two kinds of diffusion. Now we have another kind of transport called active transport, and that is when we need energy to move those molecules. And that typically is going to happen when we're moving against the concentration gradient, or we're moving from an area of low to an area of high. Now, transport proteins come in several different kinds. You have channel proteins, which act kind of as a tunnel. There's a hole in the center that allows those molecules to just pass right through. Another kind is called a carrier protein, where that protein is going to change its shape. And when it changes its shape, it's going to move the material from one side of the membrane to another. Both of these can be used in facilitated diffusion. Typically, though, we're going to see carrier proteins in active transport and channel proteins in passive transport. Now, active transport, going to need ATP for this to happen. We're moving from a low concentration to a high concentration, and this is what we call against the concentration gradient. Some examples of where we're going to see active transport in our cell is pumping sodium and potassium. There's a specialized protein pump called the sodium-potassium pump. And this is responsible for our muscle contraction and for sending nerve impulses. And in this case of the sodium-potassium pump, we want a concentration gradient. We want there to be a different amount of sodium on the inside versus the outside. So we're going to need active transport to make sure that we keep these ions at a gradient. Some other ways we can see active transport is if we want to move a large amount of materials. That is going to require energy, and the pictures we're looking at here is an example of endocytosis. So we have the inside of our cell here, the outside of our cell here, and this is our cell membrane. And we want to bring this material into the cell. So we're going to see that the cell membrane is going to pull down, and as it pulls down, it pulls the material that's on the outside down as well. And then it's going to pinch off and create a vesicle. And inside of that vesicle is the material that was originally on the outside of the cell. This process requires energy, and like I said, it's called endocytosis. Okay. Now, in endocytosis, there are a couple of different kinds. Pinocytosis is what I like to call cell drinking, and that is when we are bringing water into the cell, or materials that are dissolved in the water that's found on the outside of the cell. So if we would go back to this picture, this might be an example of pinocytosis because we're bringing in the water and the stuff that is dissolved on the inside of it. Okay, here's a nice little animation that's showing that happen. So here are the blue molecules are representing water, and then these are different materials that are dissolved in the water. This is the most common form of endocytosis, just because the outside of our cell is all fluid. It's extracellular fluid with stuff dissolved in it. So if we are pulling this down and pinching it off, we are going to have our water with our materials dissolved in it. Another kind of endocytosis is what we call receptor-mediated endocytosis. So in this case, we have proteins, or these purple things, attached to our cell membrane, and they are going to attract different molecules to attach to them. When they attach and capture those molecules, then we're going to see the cell membrane pull down again and pinch off and form a vesicle. Now, what's different from this, from pinocytosis, is that we are targeting specific molecules that we want to bring in, and we also coat this vesicle in a bunch of proteins to act as kind of extra support. Now, these molecules that we're attracting are typically going to be hormones or cholesterol, sometimes steroids, different drugs maybe. Okay, so these are going to be targeted to specific molecules that we want to bring into the cell. And then the third kind is what I call phagocytosis, or cell eating. And this is where you will have an actual kind of particle or food that is brought into the cell. Bacteria are going to use this. Amoebas are going to use this process in order to engulf their food and then be able to digest it. Here's an example or a picture. I think it's a really cool picture. Here's an amoeba. It's extending out part of its cell membrane, and here's a bacteria, and that cell membrane is going to engulf that bacteria, bring it inside of the amoeba, and then the amoeba is going to use that as food. Now, that's how we bring things into the cell with ATP. Sometimes we want to get large amount of materials out of the cell, and if we can't use diffusion, we're going to use what we call exocytosis. Okay, so we're going to have a vesicle that was inside of the cell with materials. It's going to fuse with the cell membrane, and when it does that, it releases its contents, in the case this blue molecule, and that's going to release it outside of the cell. Pretty simple. All right, so those are the types of transport, active and passive. We talked about the structure of the cell membrane. So don't forget to write a quick little summary of what we just talked about, and I'll see you in class tomorrow.